Assalamualaikum warahmatullahi wabarakatuh. My name is Siti Nosuriani binti Hussein with metric number 206079. Today, I will show you a step-by-step technique of gram staining. The main objective of gram stain is to differentiate bacteria into gram-positive or gram-negative bacteria, which helps in the classification and differentiation of microorganisms. Gram stain is a differential staining that requires the use of at least 4 chemical reagents, which are primary stain, mordant, decolorizer, and a counter stain. Crystal violet will act as a primary stain, and its function is to impart its color to all cells. Next is the mordant, which is Gram's iodine. Gram's iodine is used to intensify the color of the primary stain. The third reagent used is the decolorizing agent, which is ethyl alcohol 95%. The decolorizing agent may or may not remove the primary stain from the entire cell or only from certain cell structure. Then the final reagent, which is safranin, will be used to pin those cells that have been previously decolorized. Since only gram-negative cells undergo decolorization, they may now absorb the counterstain. The gram-positive cells will appear purple in color and gram-negative cells will appear pink in color. The material needed for this experiment are glass slide, Bunsen burner, inoculating loop, marker, staining tray, crystal violet, Gram's iodine, ethyl alcohol 95%, safranin, broth culture, and tap water. First and foremost, we need to prepare a bacterial smear. Label one clean glass slide with the name of the organism. Then, with a sterile loop, place 1 to 2 loopfuls of culture onto the glass slide. With a circular movement of the loop, spread the cell suspension into an area approximately the size of a dime. Allow the slide to air dry completely. And lastly, while holding the slide at one end, quickly pass the smear over the flame of the Bunsen burner 2 to 3 times. The next step is the gram staining. First, gently flood smears with crystal violets and let stand for 1 minute. After 1 minute, gently wash off the stain with tap water. Then, gently flood smears with the Gram's iodine mordant and let stand for 1 minute. After 1 minute, gently wash off the Gram's iodine with tap water. Decolorize with 95% ethyl alcohol. Add the reagent drop by drop until the alcohol runs almost clear. Gently wash off the 95% alcohol with tap water. Then counterstain with safranin for 45 seconds. After 45 seconds, wash off the safranin with tap water. Lastly, blot dry with bibulous paper and examine under oil immersion. As a result, gram-negative bacteria will appear pink. This is because the structure of their cell wall is unable to retain the crystal violet stain, so they are colored only by the safranin counterstain. As for gram-positive bacteria, it will appear purple. This is due to the presence of a thick layer of peptidoglycan in their cell wall, which retains the color of a primary stain.